So this is the research from my first year of a master's student at UNH, and the title of my thesis is Characterizations of Fungi Associated with the White Pine Defoliation of Eastern White Pine. As you can see, these are just some pictures documented by an aerial photo. You can see discoloration along the outer edges here, and as you can tell, it affects mainly the lower portion of the pine trees, while the upper portions are a lot more robust. This defoliation had started in 2009 and has been continuing every year since. We see defoliation of the second and third year needles. So this is an example of a twig that I found this past June, where these are the current year candles that are coming out while all the rest of the needles are defoliated. And this is a healthy white pine that I picked from the same day. So as you can tell, there's a huge defoliation difference. In 2010, we had some aerial photographies or surveys done. In Maine, we saw 60,000 acres affected. In 2012 and 13, we saw a little bit less in New Hampshire and Vermont. But not all these aerial surveys were done during the precise time of the defoliation. So, and especially being in an airplane with the top of the crowns being more robust, you can get a either better result or a worse result depending on the situation. I also work with Isabel Monk from the Forest Service, and she was who started me on this project too. Back in 2012, she started up with doing 14 data logger sites that track temperature and humidity across New England. And we have 13 survey sites along with Forest Watch, which is a program created by Dr. Barry Rock, and additional samples where these survey sites, Forest Watch, and collectors are where people grab diseased needles, send it to our lab, and we process them to figure out what fungi are actually associated with these needles. And we also have 8 tag sites. These tag sites are locations where there are anywhere from 8 to 16 trees. Half of them are high severity, while the other half are of healthy to low severity. And at these tag sites, we rate them based on defoliation and yellowing, and we do that on the 1 to 3 scale. So basically, a 1 is if the lower crown has defoliation, a 2 is if the center part of the crown has defoliation, and 3 is if defoliation is seen throughout the entire tree. From these survey sites and needles that have come in, we have so far collected and isolated out 33 different fungal species. Some of these are pathogens, most are endophytes or latent pathogens, and they range mostly from the ascomycetes, but we have a handful of basidiomycetes too. With this, we have 3 theorized key players, or key defoliators. The first one is Lyconosicta acicola, previously known as Mycus sorelli-dionesi, Lophobasidium duxii, which is previously known as Canavigella banfieldi, and Biphyzella linearis. Along with this, we have 3 new species of Septorioides-like species, which is actually another part of my project. And this, along with being a new species, its genera is actually in a new family that we're documenting. And these 2 other species, the Stagnospora and Hemiphacidium, are known, their families are known for having tree pathogens also. So with the 3 theorized key players, our main one is Lyconosicta acicola. It has recently come in to the New England area in 2006, was the first sighting up in Maine. But previously it's known as brown spot needle blight, and it can affect many pine species across the whole range. It has previously been known to cause serious defoliation in longleaf pine in the southeastern United States and on Scots pine in the northern Midwest states, but has now come to us. Although only in the north around here, we are only seeing asexual conidia. I haven't seen any sexual fruiting bodies so far. It is spread by rain splash, and its onset is in late May and June, which is the time that the defoliation is most severe. So this is why we believe this to be the theorized key player. And these are just some symptom pictures. This is the acervulus, and these are relative symptomatic needles. The next two are needle cast diseases, Lophobasidium duxii, known as Duke's needle blight or needle cast, and Bifusella linearis. These have been reported in New England since the 1900s, early 1900s, but has only been known to be on about 0.1% of the white pine population dating back that far. So we know it's around here, and we know it is causing some of the defoliation, but its onset time is different than that of Lecanocicta, as it's early in the spring. So it doesn't really time up with the defoliation that we are seeing. These are examples from the survey needles that we got in, and this is showing right now the spread of Lecanocicta acicola. The yellow circles represent DNA confirmed sightings of Lecanocicta acicola, while the orange are locations where we are seeing symptomatic needles that have yet to be processed. But we're pretty sure that, fairly certain that they are in these locations. This is a map that shows the spread of Lophobasidium duxii and Bifusella linearis. I put these two together because they show their symptoms in their sexual fruiting structures are very similar, hard to tell apart, and it is also hard to get DNA confirmed sightings of these as they're both biotrophs, so they don't grow and auger. So what we try to do is try to just scrape off the fruiting body, but that also allows us to get some endophytes into the DNA mixtures and kind of complicates our results. So the red is DNA confirmed Lophobasidium, the white is Bifusella linearis confirmed, and the purple is just symptomatic needles that could be either Lophobasidium duxii or Bifusella linearis awaiting DNA confirmation. But so the question arises now is, why are we seeing this huge defoliation where we know that Lophobasidium duxii and Bifusella linearis has been in this area since the 1900s? Why does Lecanocicta, why did it move up from the southeast or from the northern Midwest into this area? And if we look at annual summer temperatures and precipitation around New England, we're seeing increases around 1 degree, and around 9 millimeters of rain back since 1950s. Along with these 5 years, 2005, 6, 8, 9, and 11, were ranked top 10 in the wettest years in most of New England, many states, by using NOAA and airport data. So, to track the spread, to see what the exact role climate is on the defoliation in Lecanocicta acicola, we set up a dispersal experiment at the Massabesic Experimental Forest, which had a previous clear cut. In this clear cut, I created or built 10 6-foot-high spore collecting traps. Don't have a technical term for them, but made of PVC, just went to Home Depot, grabbed some things, put them together. Each of these has 4 microscope slides, which are here. These slides are sectioned off for a 10 centimeter square area. Each pole has 4 slides, and the first pole is located at 0 feet from the furthest reach of the branch of 3 previously tagged infected trees. So these trees have been tagged since 2012, so we know that they're infected with Lecanocicta acicola. The second pole is 10 feet, and the third pole is 20 feet. We also placed a pole in the center just to see how far these spores can actually spread based on rain. These slides were assessed every 4 days, along with rainfall. Temperature and humidity were also gathered from this site. So what we see here is that there's an initial spore release in early May, but peak spore release is seen mostly during the month of June, as by these little white lines here. And we can see that there's obviously a reduction as we get further out. Most of the spores land around 0 feet, so right at the tip of the branches, and decreasing as we get out. This graph basically shows it's an additive scale rather than just initial spore releases or different spore releases. So we can see when the actual peak is by using the slope. And during the month of June, we have the highest slope, and it actually starts to decrease once we get past June, which showing slight increase, slight spore release after that, but nothing too significant. This little box that I indicated here is the period where we saw the highest defoliation event during Massapequa. Like every time I went there, it'd be raining needles instead of water. So as I said, we looked at what climatic factors were really affecting Laeconocycta acicola. We really didn't see that much correlation between the average 4-day temperatures and the amount of spore release during the month of June. We saw a significant correlation between average 4-day precipitation and average spore release, but we saw even a higher significant correlation between average 4-day relative humidity and spore release. With this knowledge, I went back and started with our average defoliation severity. So as I said before, we did this on the 3 rating. I averaged the 8 sites together based on state. So we have Maine, New Hampshire, Vermont, Mass. And right here in blue, we see 2012, in yellow is 2013, and 2014 is purple. So these are the defoliation ratings based on state. And with the help of NOAA's land mapping based tool stations, I was able to create an area, find a 15-mile radius around each site to gather temperature and precipitation data. So I did that for every site. And as we see here in Maine, this blue is actually 2011. So what we were able to see is as precipitation was increasing, so was the defoliation, but it was the precipitation of the previous year. So I just did a correlation and found that defoliation severity was significant with average springtime precipitation of the preceding year. But with the knowledge of the dispersal experiment, we saw that there's even greater correlation between average June precipitation of the previous year and defoliation severity. So as a take-home, we believe that the primary pathogen of this white pine needle defoliation is Laeconocycta acicola. Relative humidity is the driving force for the current year's spore production or spore maturation. And June precipitation is the best predictor of the next year's defoliation event. Just like to thank my advisor, Kirk Broders, Isabel Monk, and the NHAES for allowing me to talk today. And if there's any questions.
So,
Yep.
I have in my forest about a 20-acre white pine portion in some old pasture.
Mm-hmm.
It's probably 70 to 80 years old. And early last summer, I saw a lot of needle drop, and it was the first time in a long time that,
Yeah.
And I'm over in Orford, south of Woodsville and Bath, I think that you had on the map.
Mm-hmm.
About 30 miles south of there.
Yeah. Oh, nice.
I don't know if it,
Well, it is widespread. We're trying to find actually locations where there's healthy pine stands. And as I said, we had these tagged trees which were high severity and healthy or low severity, but those were back in 2012. Now in 2014 and 15, those healthy low severity trees are now medium severity trees. So it is spreading, it's getting worse, and it's pretty much everywhere.